Okay, hello everybody, and welcome back. Before we start this recorded class, I just want to remind you that your grades for your test are up. And overall, I was really pleased. That was your first test in microbiology. And classes don't actually usually do that well on their very first test, but this class did. There's some A's, B's in there. Those of you who did not do well, please remember, revision is the most important way in which you learn. Repeat stuff, repeat it over and over and over again, because that's the only way that you get your brain to accept it as a memory. And these tests do require careful revision before you write them in order to do well. But many of you did really, really very well. So I was pleased. Okay, I'll also tell you that there is a new assignment up, but that assignment you may only be able to complete next week because it also covers material which is still to be discussed. And that will be discussed on Monday, probably. So this is where we ended off in our face-to-face class. We were discussing the role of ATP, and the singular role of ATP is to provide biologically useful form of energy. And that energy that contained in ATP is in fact the way in which catabolic and anabolic reactions are linked in the cell. It's ATP which provides the primary link between those processes. So for example, we begin with the brickwork of ATP, which is adenosine diphosphate. And adenosine diphosphate, it can in fact carry some energy, but we usually see the phosphorylated version of ATP being used in biological reactions. ATP is adenosine triphosphate. So what we're doing is we're adding a phosphate to ADP to create ATP. That requires a considerable input of energy, as we're going to learn later on. There has to be an input of energy in order to reduce ADP with a phosphate group. And when that energy is usually derived not from a fuel of some sort. So the energy locked in, stored in a fuel has to be accessed, liberated so that it can fund the phosphorylation of ADP. This, in fact, is how anabolic and catabolic reactions are linked together. So let's take, for example, a real common source of energy in the cell, which is glucose. Glucose is a carbon compound, and it has 6 carbons and many hydrogens attached to it. And each of those hydrogen bonds contains a lot of energy, and it is that energy which the cell is going to access. And that is accessed in the process of catabolism. Glucose is broken down in order that the energy contained in some of its hydrogen bonds can be liberated and used to phosphorylate ADP and create ATP. When that happens, that energy now has been moved. It's been moved from the glucose into ATP molecules. The ATP molecules here can now be oxidized. They can be oxidized to liberate energy. And that energy then is transferred into anabolic reactions, which are building reactions, which require an input of energy. So we store energy in some form as a fuel. We literally burn it in order to liberate the energy, to take that energy, and we put it into the phosphorylation of ADP, store it there temporarily, and then use it in anabolic reactions, which require an input of energy. Here's the molecule itself. This is ATP, and it's actually a nucleotide. We're going to encounter something very similar when we talk about DNA and RNA, but it is a nucleotide. Here's it has a sugar component here. It has a nitrogen-rich base here, and here's the critical part in terms of ATP's activity, and it's these three phosphates: mono, di, tri, 1, 2, 3. It is this energy here in this bond here, which is the third phosphate bond, which is of particular use in biological systems because it's very unstable and it can actually be transferred. It is usually transferred together with this group here, together with the phosphate group. Okay, so I hope that's clear. So this is just a summary sentence for you, but this at the bottom is really important. And that is that ATP is itself only a short-term energy storage. If we want to store energy for a long time, then we have to take off the energy and put it into a stable molecule. ATP is not a stable molecule. It's a very unstable molecule. It loses that last phosphate group together with its energy quite easily. But if we want to store energy for a long term, we have to put it into a stable compound. And those stable compounds are substances like glucose that we've already seen, and other carbon-rich compounds, which are able to bond a lot of hydrogen. Because ultimately, the energy that we're talking about is lodged in electrons. And electron together with the proton, one electron, that is a hydrogen atom. So when we look at our fuels, they usually consist of long chains of carbon, with many attached hydrogens, all of which are potential energy source. In eukaryotes, especially in plants, starch is one of the most important storage compounds. And starch, all it is polysaccharide made of individual glucose units, thousands upon thousands upon thousands of them, all joined together into very, very big molecules. But those molecules can be enzymatically broken down to liberate the energy lodged in those hydrogens, especially in their electrons. So starch is the principal product, storage product for plants. Plants also store lipids, and animal cells store lipids as fats, that sort of thing, but also as glycogen. And glycogen, here is a glycogen molecule, very, very similar to starch, but it is the typical storage product for animal cells. And it's extremely abundant in your liver and also in your muscles, which need a ready source of energy for obvious reasons. So glycogen is an animal product, starch is the equivalent plant product. And both of them will also make lipids, oils, and things like that. In prokaryotes, which is our principal interest, glycogen is a common product. Sometimes glycogen molecules get linked together. And glycogen is just it's a polysaccharide, as we've said. But if you link enough of the molecules together, it actually precipitates out and becomes visible. And you can actually see glycogen crystals inside prokaryote cells. The other type product is one called polybeta hydroxybutyrate, PHB. And that is another prokaryote storage product. And they also make lipids as well. So, all right, so we need to just talk a little bit about really basic chemistry and about why reactions take place at all. If I want to get two substances to react with one another, I have to do two things. The first is that I have to bring them into proximity with one another. That's step one. Step two is that they have to meet with sufficient energy that they collide, and the energy of collision is sufficient to reshuffle the bonds. And you end up with two products from the reaction. You end up with products from the reaction. A simple example is given here. This is methane. Methane is the simplest of all the organic molecules. It's a carbon together with 4 hydrogens. That is household gas. Okay. And if household gas, if we want to use it, we have to burn it. We have to burn it together with oxygen. So those are our two reactants, methane and oxygen in this particular reaction. Those two molecules have to collide sufficient energy that they actually reshuffle the bonds, and they end up producing carbon dioxide and water. And in the process, they liberate energy. There's energy locked into these bonds of the hydrogens to the carbon. That energy is liberated in the process of burning to produce carbon dioxide, water, and energy. We see, when we burn household gas, we see it as light energy, but we most importantly feel it as heat. So that's laboring the point is this is a simple reaction, it seems like, but there are some important things to note about it. The first is that those molecules have to collide with sufficient energy for the reaction to take place. If they don't have sufficient energy, they can be close to one another and nothing is going to happen. And this is the basis of collision theory. That in order for a reaction to take place, the substrates must collide with sufficient energy. And that energy of collision is called the activation energy. They require a certain input of energy. Something has to speed them up so that they collide with sufficient energy. So that is the activation energy. And we observe the progress of these reactions by observing the reaction rate, the rate at which these reactions take place. So some reactions take place extremely rapidly. Others take place extremely slowly. And we're going to find, in fact, that in most catabolic and anabolic reactions, especially in the cell, in living systems, take place very, very slowly under normal circumstances. So the reaction rate is critical, because if the reaction rate is too slow, it's not going to be useful. And especially in a living system, if the reaction rate is inherently very low, it may take an enormous amount of time for a useful quantity of product to be produced, for example. So there are various ways in which the reaction rate can be altered. The first is that we can increase the proportions of the reactants. We can increase the concentration of the reactants. That increases their proximity to one another and increases the likelihood that some of them are going to react with one another. We can also increase the reaction rate by raising the temperature. That is because if you raise the temperature, you increase the kinetic energy of molecules, so they collide with greater force. We can also increase by increasing the pressure, forcing them close to one another. But in biological systems, the most important way in which we increase the reaction rate is by the use of enzymes. So this is a really simple explanation. I think in your text it goes into more detail, but this is a simple explanation. of the role of enzymes. There are many substances which will participate in a reaction, which speed up the rate of reaction of the reaction, and yet they emerge, they are unaltered by the reaction itself. There are many inorganic substances that do this. The one that may be familiar to you, for example, is platinum. Platinum is used as a catalyst, for example, in exhaust pipes. And that's its role. It speeds up reactions inside the exhaust pipe to limit emissions. So a catalyst is something that speeds up the rate of chemical reactions, but it itself is not altered by them. It emerges afterwards unaltered. Enzymes are biological catalysts. They are substances which speed up biological reactions, but they themselves can emerge from the reaction at the end unaltered and ready to perform another catalysis in the reaction. So what we're going to look at, we're just going to take a very simple example in this figure. We're looking at a single reactant. It's molecules which are bonded together in this fashion. And we're going to watch as the bonds here are broken, and we end up with two separate products at the end. For this to happen naturally, what is probably going to happen is we have to increase the kinetic energy of these molecules or whatever, and so that those bonds would break spontaneously. That would require a considerable input of energy, and that is the activation energy. It's displayed here. If we were watching this reaction take place, we'd have to put in a considerable amount of energy for this reaction to begin to go spontaneously. And here it goes. Here we put in the amount of energy required, the activation energy, and then the reaction proceeds. Now, in biological systems, it's very difficult to add heat or anything to the system in order to speed up reactions. Heating up is not an option. Instead, biological systems rely on enzymes, which are biological catalysts. And what any catalyst does, and enzymes do it, is to lower the energy of activation. So here's the activation energy without enzymes. It is considerable. With an enzyme, the activation energy is lowered, and it is lowered so much that it makes the reaction much more likely to occur, and the reaction proceeds much faster than it does without. So here is the activation energy with the enzyme, much lower than without, and the reaction proceeds. So that is the principal activity of enzymes. They lower the energy of activation for reactions, and they speed up the rate because they make the reaction much more likely to happen. They do so in specific ways. So first of all, enzymes are incredibly specific. They are specific for the substrates that they are going to act on. And their specificity is determined by the fact that they have only one small portion of the molecule which interacts with the substrate, and that is called the active site. We're talking here, 99% of the enzymes in living systems are proteins, very complex proteins, and often very large. But they have a small area called the active site, which binds the substrate. And when that substrate is bound, here we're going to have a look here. This is sucrose, and this is an enzyme, it's variously called invertase or sucrase. As soon as you hear ASE, you know you're dealing with an enzyme. Okay, so this is sucrase or invertase here. You can see here, diagrammatically, they show you, here's the shape, and that shape fits the shape of the substrate molecule. So when that binding takes place, that forms a complex. It's called the enzyme substrate complex, and it's the essential first step in the catalysis of the reaction. Once the active site binds the substrate, the enzyme shape actually changes, and it's the enzyme shape change is what catalyzes the reaction. For example, in this case, the enzyme actually flexes out, and it's bound tightly to the molecule. The molecule consists of two parts, and each part has its own binding. The enzyme flexes and it strains the bonds between the two parts and snaps them. Once that has happened, the enzyme loses its affinity for the substrate. It's no longer the substrate that the enzyme interacts with, and it releases the component parts. It liberates them. And here they are. Here's one molecule is fructose, and the other is glucose. That's what makes up a sugar molecule, sucrose molecule. So this is a simple example of how an enzyme will operate. In other cases, the enzyme may bind to reactants separately and into this enzyme substrate complex. And that binding, again, induces a shape change, which may force the molecules together and press them into reacting with one another. So they don't now need to have kinetic energy to smack into one another spontaneously. Instead, the enzyme does it. The enzyme forces them together. Enzymes have various ways of catalyzing reactions. Sometimes, for example, they will add a hydrogen, they will add energy to substrates in order to drive a reaction in a particular direction. But in all of these reactions, we start with the enzyme substrate complex, which undergoes changes to induce a reaction. And then the enzyme and the products are released at the end of it. Okay, so here's the, just to show you, this is what sucrose looks like. And there's a glucose molecule and then a fructose molecule. And the enzyme we've just seen, invertase, cracks that and opens it. Now, and releases glucose and fructose. Now, let's go back here. Now, can you see? If I take sugar, if I take table sugar, I can leave it in the sugar dish on the counter for thousands of years. Some of those molecules in the sugar dish will spontaneously break up. They may get hit by a cosmic ray or something like that. They may be hit by a very fast moving oxygen molecule. Whatever, there is a very low rate of reaction because sugar is extremely stable. It requires a very big input of energy for it to spontaneously break into its component parts. But with the enzyme invertase, it happens quite easily because the enzyme itself provides the catalysis, which will result in the reaction taking place at a much lower activation energy. Okay, so enzymes are a science all of their own. They're extremely complex substances, and they often don't operate simply with the protein portion. Very often they require other added parts before they become active. And that's actually quite useful for the cell because it gives the cell an on-off switch. as well. If you activate an enzyme, you put in a part which allows it to perform its catalysis. So there are various substances which will be part of the final enzyme, active enzyme. The protein part of the enzyme here is called the apo enzyme. Now, I told you 99% of the time, enzymes are proteinaceous. There's one important group of enzymes, however, which are RNA based, and they are called the ribozymes. So remember, almost all enzymes are proteinaceous, but RNA also has catalytic ability. But in case here we're going to just talk about the proteinaceous enzyme. This is the apo enzyme is the protein part, and it may have associated with it a cofactor. So the cofactors are many and various. Vitamins, metal ions, and organic substances, which are essential to the functioning of the enzyme, which we call the coenzymes. A cofactor is something that must interact with the enzyme in order for it to be activated, in order for it to operate. A special subcategory of cofactors are the coenzymes, and they are organic substances. So, for example, many of the essential vitamins, for example, are cofactors. And we're going to encounter several other kinds of organic substances which are coenzymes. So here is another little aspect of our studies of energy inside the cell. We've said that energy is usually in biologically useful form, which is ATP. So that's fine. There's the energy, and we've hinted already that it involves the electrons, reducing it and storing energy in the ATP. The question is, we keep on saying, well, we're going to transfer electrons. How? How do we transfer? We can't put in wires inside the cell. And we can't have electrons just flying around willy-nilly inside the cell. They just don't do that. Instead, electrons have to be carried. They have to move from their source like a fuel. If we want to transfer electrons, we've got to allow them to bind to an electron carrier. And that forms a reduced substance, and we refer to this as reducing power. And those electron carriers are extremely important in almost all of the reactions that we're going to talk about of transfer of energy. So the first one is called nicotinamide adenine dinucleotide. That is NAD, and in its raw form, it is ionized. It is positively charged. Okay, so in its raw form, it actually has lost an electron. It is positively charged. The other one, nicotinamide adenine dinucleotide phosphate, very, very similar, but it occurs predominantly in plants. The other one is flavine adenine dinucleotide, FAD. And a third one that we're going to encounter later on is called coenzyme A. And remember, coenzyme, that is an organic cofactor. So these are actually based on nucleotides. The nicotinamides and flavin, these are based on nucleotides. And coenzyme A is a separate substance. So here is our apo enzyme, inactive. It has to bind to its coenzyme, which may be any of these. It has to bind to its coenzyme here before it becomes active. Now its active site is complete and it can bind the substrate. So here's the important thing. We're going to see that very often in biological systems, the coenzyme is one of these electron carriers. So for example, if we're going to produce a substance called NADH when we add electrons and a proton to the substance. So we're going to put in here a reduced form of our electron carrier. But the characteristic of these electron carriers is they get oxidized and reduced very, very easily. So what we've done is we've got our enzyme, we've put in here a rich source of electrons and their attached energy. And now we can bring in a substrate and we can perform a reaction now, which requires an input of energy, further input of energy. And that is catalyzed by the apo enzyme. So it's a complex process. These substances interact with one another. And they're interacting so that the transfer of energy can be directed and it can be facilitated by the enzyme. Remember that the electron carriers, we'll talk about it again in a while, their exact function. But this is the responsibility of electron carriers to actually temporarily bind electrons and transport them to where they will be useful. We can't just have electrons flying around willy-nilly. Okay, so the activity of enzymes is itself affected by various factors. And the most important one probably is temperature. But pH and the substrate concentration, these are all important things as well. And enzymes can actually also be inhibited. Their activity can be slowed down. And this is really important in the cell. Okay, let's have a look at the impact of temperature. Here's a typical graph showing activity of an enzyme at various temperatures. And you'll immediately see that there is a temperature where it works best. Enzyme activity is at its peak around about here. And unsurprisingly, that is around about, for this particular enzyme, that is around about 37 degrees Celsius. Well, what is our body temperature? Our body temperature is right around there, about 37. And many bacteria, especially those which are commensal with us, all those which are pathogenic, have their temperature optimum for their enzymes is also around about 37. It depends on what organism you're talking about, and it depends on where that organism lives. There are other bacteria, for example, which grow at very, very high temperatures, or they grow at very, very low temperatures. And their optimum might be different. That's what this is called. It is the optimum temperature. So if I cool, for this particular enzyme, with its optimum 37, if I cool it down, what I'll see is that the activity will decrease. It'll decrease and decrease. And then there's a temperature, a critical temperature, at which activity will stop. In this case, about 10 degrees Celsius for this particular enzyme. If I warm this enzyme up, it is true for most enzymes, not all, but for most enzymes, if I cool them and heat them up again, their activity is restored. So warm it up and it will go back up and be active at its optimum. But look what happens when I take the enzyme above its optimum. The energy, activity very, very rapidly decreases. And if I cool this enzyme down again, very, very seldom will an enzyme be restored. This heating above the optimum is very frequently lethal to the functioning of that enzyme. And that is because most of these enzymes are protein. And the whole protein structure becomes altered when we raise the temperature too high. We say that the protein is denatured. The bonding structures which hold the enzyme together in a particular shape, creating its active site and everything else, all of those bonds are shattered, broken, and new bonds may be made. The enzyme is denatured, it is killed by high temperature. There are bacteria which have very, very high temperature optimums, up near boiling point in actual fact. So their enzymes would be specially adapted. Okay, pH also has an effect on the activity of enzymes. And that is because most of the bonding, a lot of the bonding, in the protein creating its shape is actually through fairly weak but very common bonds like hydrogen bonds. And those bonds are very much affected by the pH. And the pH is a measure of the number of protons that are in solution. And protons are positively charged. And if we have excess of those protons floating around, for example, then you may end up breaking all of the hydrogen bonds in the protein. Same if we if we make the solution extremely alkali, then the same thing happens. We end up breaking and distorting the bonding structures which hold the enzyme in its critically important shape. So here, have a look here, and you'll see here is an enzyme with its optimum, its pH optimum is about 7 there, which is the commonest. Most bacteria, for example, have a pH optimum around about neutrality. Sometimes a little bit low, or a little bit lower, or a little bit higher, but very seldom far away from neutrality. There are specially adapted bacteria which are adapted either for very low pH, very acid conditions, or for very high pH, or highly alkaline. But most have an optimum around about neutrality there. The third thing which drastically affects the rate at which enzyme activity occurs is the concentration of the substrates. So, for example, you just picture the enzyme is only going to be performing the catalysis when it's bound to its substrate. If the substrate is extremely rare, we may have many enzyme molecules which are simply unable to bind substrate, and so nothing happens. Low concentration of substrate, enzyme activity is usually very, very low. As we raise the concentration of the substrates, more and more of the enzyme molecules are able to bind substrates and perform the catalysis. So what we'll see is that the enzyme activity rises. But there reaches a point where we have so much substrate present that all of the enzyme molecules are occupied. And after that, adding more is not going to speed up the rate of reactions because all of the enzymes are occupied. And so we see this typically level off at a certain concentration of the substrate, as you see here. So it rises in this typical curve, and then it plateaus because here all of the enzyme molecules are occupied. Okay, so there are also ways of controlling enzymes by shutting them down, for example. And this is called inhibition. And we're going to discuss two kinds of inhibition, two kinds of inhibitors. The one is a completely natural and frequently used, And this is called non-competitive inhibition. And that is very frequently seen in cell. The other is called competitive inhibition. And it's a completely artificial situation. Competitive inhibition is typical of a poison or a toxin, which interferes with the enzyme and stops it from working. It's an artificial situation. It's not used by the cell as a way of controlling enzyme activity. So here, for example, let me just head out there. Here, for example, is an enzyme. Here is its active site, and in competitive inhibition, the inhibitor binds to the active site, often just to one part of the active site, and it blocks the active site there. And the substrates then are unable to bind. This is usually a permanent situation. This is usually lethal to the activity of the enzyme. And this is true of many poisons that do this. And probably the most famous one is cyanide. Cyanide binds to the active site of one of the respiratory enzymes that we're going to see when we talk about how cellular respiration operates. And cyanide binds to one of those enzymes and stops respiration from taking place. But remember, this is completely artificial. It's not used by the cell to control enzyme activity. The other kind of inhibition is very frequently used by the cell. And in fact, most metabolic pathways have at least one of their enzymes, usually the very first enzyme in a pathway, which the cell can control. It can switch the enzyme off by this particular kind of inhibition, non-competitive inhibition or allosteric inhibition. And in fact, I prefer the term allosteric inhibition. It describes it much more succinctly. So here's how allosteric inhibition works. Here's an enzyme, and we've got the active site here. It's the same enzyme as here. Here's the active site that our substrate would bind to. But elsewhere on the enzyme, there is another site. It's not an active site, but here it is here, and it's called the allosteric site. And it will bind a substance. If the substance is in high enough concentration, the allosteric site will bind that substance. And when it does so, it changes the shape of the active site. It distorts the molecule. And the active site can no longer bind to the substrate. This is not a permanent situation. This is temporary. The inhibitor can come off there and the enzyme will restore and be able to proceed with the reaction. And this is a very clever way in which the cell can actually control the activity of enzymes. It can provide enough of the allosteric inhibitor that it will bind to many of the enzyme molecules and will begin to slow down the overall rate of the reaction. And we're going to see this typically in the very first enzyme of a metabolic pathway. And surprisingly enough, the inhibitor in that case is very often the product of the metabolic pathway. The reasoning is that as the concentration of the substrate, of the product, rises, there is less and less need for the pathway to operate to produce it. And so the product itself ends up inhibiting the enzyme at the start and shutting down the whole pathway. When the cell's obviously making that product for a reason, it's using it. As the concentration of the product drops inside the cell, so the enzyme becomes activated because the inhibitor is lost. We'll see, I'll explain this again, but remember the two differences. This is permanent, and poisons the enzyme. This reaction here is not permanent. In fact, it's very temporary, and it can be used, the allosteric inhibitor or non-competitive inhibitor can be used to slow down the activity of enzymes. Okay, that system is called feedback inhibition, where we shut down a pathway as the product accumulates. And here is an easy explanation of it. Here's a pathway. So a metabolic pathway, we've seen before, a metabolic pathway is a series of linked reactions. We break down an overall reaction into steps. And each step has its own enzyme in the pathway. So for example, here is the very first enzyme here. And it is going to operate binding the substrate there. It's going to release a product, which is an intermediate. And the intermediate is acted on by a separate enzyme, enzyme 2. That produces a second intermediate, and that is operated on by the third enzyme, enzyme 3. And we end up with an end product of some sort. So how is this pathway now going to be controlled? Well, this very first enzyme has an allosteric site. There it is there. And if we allow an inhibitor to bind there, we're going to alter the shape of the enzyme. The enzyme will no longer be active. If the enzyme 1 is no longer active, it can't produce an intermediate. If it can't produce an intermediate, none of these have a substrate or are going to have a substrate to work on. By shutting down the first enzyme, we shut down the whole pathway. So here is the end product here. And in fact, the end product is the inhibitor. As the concentration of the end product rises inside the cell, the product actually binds to the allosteric site and shuts down the pathway. It inhibits the whole pathway. But now the cell is using the product. It's using up the product of the pathway. Remember, I told you this is a temporary binding. In fact, it's very temporary. And this continually leaves and gets replaced by another molecule of inhibitor, as long as the concentration of this product is high. When the concentration of this product falls to low levels, then this enzyme spends more time being active than being inactive. If the product is in high concentration, the first enzyme will spend more time inactive than active, purely by this switch of binding here, of non-competitive binding. This is the typical system for feedback inhibition. And we'll see this over and over and over again. It is extremely common in metabolic pathways, and it's one of the principal ways in which enzyme activity is controlled inside the cell. There are other ways to control enzymes as well. But those are usually by controlling, for example, we saw the coenzyme binding, which actually often phosphorylates the enzyme and makes it active. There are ways to control that as well. But feedback inhibition is the most important way of control. So I'll just finish off by telling you, I said before that 99% of enzyme activity, 99% of the enzymes are actually proteinaceous. But it is really important to remember that RNA, which is a nucleic acid, also has enzymatic ability. Certain RNA molecules have enzymatic ability. And the most important one that we are going to encounter is the enzymatic activity of RNA inside the ribosomes. Ribosomes are responsible for protein synthesis. And that the activity of the ribosome is controlled by a set of RNA enzymes or ribozymes. So but there are many of these ribozymes which operate in all sorts of different areas of cellular activity. Okay, so I'll leave that there, and then I'll see you on Monday.